I'm Deb Newberry, and I'd like to talk to you about biomaterial scaffolds. What in the heck are those? We'll get to them. But let me tell you just a little bit about myself. I'm kind of a science nerd. And, if you ask my children, they will probably agree with that definition of who their mom is. In fact, when I was in second grade, I told my parents I wanted to be a scientist. And I was very blessed because they allowed me and they supported me in that process throughout my whole career. When I went to college, I couldn't decide exactly what science I wanted to be involved with, so I took 3 majors: physics, chemical engineering, and mechanical engineering. I was even a pre-med major for a while because I couldn't make up my mind. I ended up deciding on nuclear physics as the major area of my study. And when I started working, I ended up doing radiation and thermal analysis and testing on satellite systems. In fact, at one point in time, there were 12 satellites in space that had my stuff on them. Over the last few years, I've also gotten involved in biotechnology. It's an easy thing to do because what's happening today is all the normal columns between physics, biology, chemistry, all of the engineering, they are starting to join. And that's one of the challenges for both students and educators and industry, because we're seeing such multidisciplinary aspects in so many of the standard traditional disciplines. And so what I'd like to talk to you about is biomaterial scaffolds, as I said. And for decades, scaffolds have been looked at as a way to improve healing, to speed up healing. For example, our bone cells are long, narrow cells, and they take a long time to come together to finally heal a bone break. In fact, that's why it takes bone so long to heal. Why we have to wear a cast for so long, because it just takes time for those cells to come together to where they're strong enough to support weight. Same thing is true for a lot of surgeries: heart, kidney surgery, back surgery. All of those cells take a while to come back together and heal. And so since the mid-90s, a lot of researchers have been looking at ways that we can help these cells find each other and heal. And so they've been looking at scaffolds. Originally, people were looking at carbon nanotubes as a scaffold material. Carbon is biologically compatible, so making a scaffold out of carbon nanotubes would make a lot of sense. However, the problem with the carbon nanotubes was it was hard to get them to form the exact structure of a scaffold, the ladder type structure. Sometimes carbon nanotubes don't exactly want to behave themselves. The next step was to actually use DNA. The 4 amino acids that make up our DNA: A's, C's, T's, and G's. Creating a scaffold similar to the DNA structure, that ladder, that twisted ladder type structure that we're probably familiar with. Well, the problem with DNA, one of the attributes that makes it so good is that it's extremely flexible and foldable. That's what makes DNA and allows it to form the RNA that we need to create proteins and amino acids and all the structures of our body. But that flexibility also made DNA not perhaps the best scaffold structure. So more recently, they are looking at proteins, combinations of proteins, I'll just abbreviate it, and amino acids as a biological material to create scaffolds out of. Researchers have figured out ways to structure the protein and the amino acids to make them into a physically viable structure. Now, if these materials have to work with the body, they have to be able to deal with the water environment that we have in our cells. They have to have a certain property in terms of hydrophobicity, being able to interact with water, or in some cases, hydrophobicity, which means that they aren't going to interact with water, because all of our cells and the liquid in our bodies are based on a water type structure. So these scaffolds have to have a certain ability to interact or not interact with the water, depending upon the type of cell structure and actually the function of the scaffold. So they have certain properties that are related to how they interact with water. They're also going to have certain physical properties. Their ability to stay stable, their ability to interact and interface with the cells that they want to support. So there are certain material properties. Maybe it's density, maybe it's strength, stress, the ability to deal with stress and strain, the physical properties. So there are very unique properties that these biomaterial scaffolds have to have. That's really nice because it makes it work well with our body. However, because of this specificity, they become extremely expensive. And the testing as well as the manufacture and fabrication can be extremely expensive. This is kind of an aside, but one of the issues that has to be addressed when you're creating biological materials, whether it's a vaccine, whether it is a drug or a nanoparticle or a cancer treatment, you always have to make sure that that entity that you are creating meets the specifications for quality. That's a good thing. That's always a good thing. But part of the problem is, how do you test something without actually destroying it? You know, a lot of times when you want to test to make sure that your drug has the properties that it has, that it's going to interact with the protein, the pieces of the protein that it's supposed to interact with, and not interact with the pieces of the protein that it's not supposed to, because that interaction is going to cause the side effects that you don't want. But in the testing of that drug, in a lot of cases, the very testing destroys the drug that you're trying to create. So that's one of the challenges of testing and quality assurance in the biotechnology field, is how can we create a test to make sure that the entity that we created has the properties that we want it to have without destroying it. That's a big challenge and a significant challenge. Also, for you mathematicians out there, there's a lot of statistics involved in this. Probabilities, assessment of the risk. How much of a sample do you have to test to make sure that the sample as a whole is good? Those are questions that mathematicians need to answer. So we've got a scaffold that's going to have special, very defined properties, whether it's how it interacts with a cell, what it's created, what its composition is, how it interacts with water, or how it has interactions with certain physical properties. Because of that specificity, it becomes a very expensive product. Scaffolds are a very expensive product to create, to use. Not necessarily the best thing. So what do you do about that? Well, what I would suggest is that you look at these properties and step back from biological scaffolds, biological applications. Think about other places in the world, whether it's building a house, whether it's creating windows, whether it's building a car or a new machine, whether it's creating a new smartphone or a computer processor. Are there other places, other markets, other products that might require and need exactly these properties? And as you start to expand your vision outside of the biotechnology, outside of the biomaterial scaffold application, you start seeing places where those physical attributes or that interaction with water might be a benefit. By looking at those other market segments, two things can happen. One, you can expand the market segment to create more products, and by building more products, the price of each device will go down. Each scaffold will be reduced. You're making more of them, selling more of them. The second thing that's most important is that in expanding your field of view away from biology, away from biotechnology, and increasing it into physics, chemistry, engineering, mechanical engineering, crystallography, you've all of a sudden expanded your field of knowledge. You're pulling in different people, you're interacting with different people. You're maybe expanding your own field of knowledge by thinking outside of the box. That is one way to drive the price down, increase your revenue. Think about it. Next time you see an article, whether it's on the internet or in a magazine or in a newspaper, think about the properties that that particular entity requires, and then think about ways that that property might benefit another industry, another product, or another person. Thank you very much for your time.